So I want to ask this question, how might we use big data thank you very much, how might we use big data in Alzheimer's disease research? And big data is all over the place at the moment, isn't it? It's certainly in the papers in the UK, but I am guessing it's pretty much in the papers wherever you are as well. It's certainly something that is in the zeitgeist, as they say. Next slide, please. And there's all sorts of responses that people have to big data and artificial intelligence and machine learning and these kind of things. One is that we're getting such huge amounts of data that it's just simply overwhelming. Another is that, this is the more optimistic view, of course, that big data is power and is going to transform the way that we live. And the third, which is perhaps the most current at the moment, is that big data is very worrying, that there's lots of problems around big data, and the whole Facebook, Cambridge Analytica, Donald Trump, Brexit, pretty much everything bad in the world is being blamed on big data right now. So this is an interesting field to be involved in. Next slide. But actually, big data isn't any of the things that I've just described. It is technically described as these three things. It's data that has volume, variety, velocity, and complexity. And I apologize for the very UK or even London-centric slides that I'm illustrating this with. Volume is obvious. Big data is voluminous. There's lots of it. But it's important to remember that big data is not just about very large data sets. It's also about data sets that have a great deal of variety. So they're data sets where you have very different kinds of data in the same context, in our case, in the analysis for Alzheimer's disease. Big data is also defined as data that is changing very rapidly. So it's data that comes at you with great velocity. And because of its volume, variety, and velocity, big data is very complex, difficult to understand what it means. It's difficult to determine signal above noise. Next slide. So what I'm going to try and do is to just illustrate how we might or might not use informatics and intrinsically big data informatics in the drug discovery pipeline from phase 0 to phase 4. Next slide. I think it's a build now. So just in ordinary I know you know this, but just in ordinary layperson's terms, phase 0 is about finding a target and making a drug. Next slide. Phase 1 is about safety. Phase 2 is about proof of concept, meaning is it a good idea to find a drug for this particular target. Phase 3 is, does this drug actually work for patients? So it's the most important part of drug discovery. And phase 4, next slide, not to be forgotten, is does the drug work in the real world as it seemed to work in clinical trials? And the answer to that is often it doesn't. So we need to do those studies both for efficacy as well as safety in phase 4. Next slide. So I'm going to talk a little bit about how we can use big data. And in particular, I'm going to talk about how we can use data that we already have. And I'm going to start by describing some platforms that enable us to use data that we already have for these studies from phase 0 to phase 4, very pertinent to EPAD, which was constructed in a way that EPAD was constructed in a way that EPAD was itself going to be facilitated by the platforms I'm going to describe. Then I'm going to talk about how we can reuse both molecular and clinical data in drug development. And then I'll think a little bit at the end about how we might go forward in the future. Next slide. So starting with platforms, the sister program to IMI-EPAD is IMI-EMIF, the European Medical Information Framework. This is very like EPAD in terms of its organization. A very large number of academic partners, some SME partners, and partners from the European Federation of Pharmaceutical Industry Association, FPIA, and you can see them there. And I want to note that Janssen J&J is the co-lead together with me for EMIF program, Bart van Nieuwenhuizen, and Synapse has played a critical role as an SME partner in much of the project management. So 58 partners, 14 European countries, 56 million. Those are the figures. Next build. Our goal, next slide please. Our goal was to become the trusted European hub for healthcare data. So importantly, it wasn't to do studies in Alzheimer's disease, although as you'll see, that was one of our use cases along with metabolic disorders. It was essentially to set up a platform, and I think it's fair to say we've been successful in doing that. That platform was to help you, the researcher, discover, assess, and reuse data. Next slide. Next slide, thank you. So one of the most important things that EMIF has done, which I think is really critically important to EPAD Academy members, is helping you to see what data is available. So the first problem that a researcher has is, I've got an idea, I want to get some data, I have no idea where that data exists or how I go about finding it. Now, when I started, I don't want to do the sort of your granddad's anecdote kind of thing, but it feels like it was that long ago. When I wanted data, I had to go to the library and look up on a piece of paper, read a journal article and try and guess whether they had the right data. We've come through 2 or 3 waves of enhancements since then, and I think that the EMIF-catalogue is the latest iteration of that. So if you wouldn't mind just building through this slide as I'm talking, what the catalogue does is, first of all, give you an overview of Alzheimer's disease studies in Europe. Next bullet. And it allows you to, you go on the catalogue, next one please, you can go on the catalogue, the URL is up in the top right of your screen, and it will give you detailed information about every study that is on the cohort. Next. It will allow you to do advanced search techniques, so you can use Boolean type search characteristics, you can combine the different variables that you're interested in. So it might be that you're just interested in MCI in people under the age of 60, where you can find the cohorts across Europe that have got that kind of data. Next slide. And it allows you to, much as if you're going on to Amazon, if you scroll down your Amazon screen, it will do a compare and contrast of different varieties of the thing that you're trying to buy. You can go onto a car website if you're buying a car and look at the fuel consumption of different cars of different makes and so on and so forth. You can do much the same thing comparing cohorts across Europe now. Next slide. And then maybe another one. So that's the key thing, but accompanying the catalogue, EMIF has got a whole series of tools that allow you not just to discover data, but to assess, access, and reuse that data. And the bit that is of most relevant to the EPAD Academy, I would suggest, is the bit in green. This allows you to go into the cohort, cohort catalogue, to select cohorts you're interested in. It allows you to determine which participants you might be interested in, for which variables, and something called the switch box is a harmonization process that allows you to put together variables from different cohorts and actually aggregate them. Now, we've already done that for a growing number of cohorts, and we've put that data on the Transmart environment, which is essentially a platform for putting data that allows you to put multiple different cohorts on, to harmonize the variables from those cohorts and to analyze them. There's all sorts of analytical tools, and you can do that in a private remote research environment. The bits in blue are broadly similar, or at least analogous, but now looking not for cohort data, but for real-world clinical data, electronic medical records in effect. And the key part of that is a common data model, which is called OMOP, that is part of the Odyssey, that's OHDSI community. And as you'll see later, a new IMI grant has just been awarded, and that's being led by Peter Reinbeck from Erasmus, and it includes many groups, including, I'm pleased to say, Oxford, that will continue much of this work for the next 5 years. Next slide. So this process allows you to find and reuse data, and this is an account of the amount of data that we've got. So if you look at the table on the left-hand side, those are the cohorts that we have available. Actually, this is out of date now. We've got something over 100,000 participants in cohorts that are on the catalogue. And if you look at the breakdown, a very large number of these people have either normal cognition or mild cognitive impairment. So they're just the sort of people we want to investigate in studies like EPAD. And if you look on the right-hand side, you'll see that we can access electronic medical records from a little over 60 million Europeans. And I'll show you some of the studies that we're doing on these kind of data sets in the second half of my talk. Next slide. So the tools that we've developed in EMIF have been used in other studies. Here's an example from Dementia's Platform UK in the UK, obviously, and this differs a little bit from EMIF in that it has very large epidemiological type cohorts in it. So as you can see here, 1 million people from the Million Women study, 500,000 from UK Biobank, 30,000 from Cognitive Function in Aging study, CFAS, and so on and so forth. Next slide. I now want to turn briefly to illustrate a system for accessing electronic health records. This is the one from the UK that I lead, but there's many others across Europe. Clinical Records Interactive Search is a system that allows you to access EMR data from mental health and dementia in the UK. So in the UK, dementia is treated by psychiatrists. It's a little different from most of the countries that you work in. And because we have an NHS, everybody that gets planned dementia care is treated by an NHS trust that is part of this mental health and dementia part of the NHS. And there's, I think, about 40 to 45 of these trusts in England and Wales. And we've put this system that we've generated on about a third of those, which gives access to 2.5 million individuals that have received mental health or dementia care. Next slide. This is a fairly simplified version of a complex system. I don't really want to talk about this very much other than to say all of the data stays firmly within the NHS trust where it was developed, but we've generated a system that allows federated access to these NHS, to either an individual NHS trust or to multiple NHS trusts, and all of this data has been mapped to the common data model, OMOP, that I talked about just a moment ago. Next slide. So those are two examples, EMIF and CRIS, of how we're accessing research data and real-world observational data, medical records data, for a variety of indications, including potentially Alzheimer's disease research and including, hopefully, research that is relevant to EPAD. So how can we actually use this data? I'm now going to give you two examples of big data studies that we've recently done in phase 0, and then I'll walk through the rest of the phases in a little bit less detail. So the first bit in phase 0 will take longer. And here we're using data that is very variable data. So I'm going to give you two examples where we've combined molecular studies with real-world studies to nominate a target for an Alzheimer's disease development. And by target, I'm using it in the drug discovery sense. And what I mean is a molecule against which we want to generate a drug. Next slide. So, you know this. We want to find targets that are part of this pathway that goes from something to do with amyloid to something to do with tau to the cognitive loss that Augusta Dieter suffered from. Next slide. So in work that I've been doing over the past decade or so, we think we've found a target. I'm going to have to ask you to take my word for that. But if you want to read a paper, there's a paper that describes the early part of this work. The detail of it really doesn't matter for the purpose I'm describing now, but we think we've got a target that is where that red arrow lies. It mediates the effect of amyloid on neurons and then transmits the Alzheimer's disease pathology. So if you take my word for it, we've found a molecular target. Next slide. How can we develop that a little more? Well, the first and most conventional way of doing it is you develop a drug screen approach, and we've done this. And in fact, I've just come from a meeting where we're extending that drug screen process. So I'm just going to say a word about how we developed a drug screen. First, we developed a high dimensionality imaging roboticized automated screen. What you're looking at here are neurons in vitro, which we've stained with a synaptic protein. So you can see the control cells in the top left, lots of synapses. You can see down in the bottom left, when we treat these neurons with amyloid, they lose synapses. And then top right is a control, and bottom right is when we treat with amyloid and we knock out the target with a inhibiting molecule, siRNA. So what that slide shows you is that amyloid damages the neurons, you lose synapses, and when we take out our target genetically, you prevent that from happening. So this is a proof of concept of a screen. And we developed this screen so that we can do this experiment without too much trouble in perhaps tens of thousands of times. We have actually done it about 100,000 times, as I'm about to show you, and we're working up to do it in maybe 500,000 times, the same experiment. So it's all done with, it's basically a big data study in itself. Next build. So what we did using this screen, using robots and doing it automatedly and analyzing it with automated image analysis, we screened 90,000 compounds. We found over 100 compounds that do the same as number 4 that you're looking at here. We took those through secondary screens. In other words, we got a person to do it, a rather more complicated screening assay. We found 10 compounds that passed that, and we are about to put the first of those compounds through animal studies in the next, I hope, 6 to 8 weeks or so. Next slide. So that study took about 4 years, and it cost in the region of, what would that be, 6,500 euros. That's about what it costs. That's fairly standard for these kind of approaches. But I told you we'd found a genetic target. So we had the genetic signature of Alzheimer's disease in that study that I asked you to take my word for. So as well as doing this conventional screen, which included some big data analytics, we did a truly big data analysis independently. Next slide. And for this, we use something called the connectivity map. It's a great study. It's a study that you can do this yourself. It's all open access. It's available at the Broad Institute in Boston. Basically, some very clever people had a great idea, and funded by the NIH, they took some human cells, they exposed those human cells to a drug, and they did this 1,500 times, so with 1,500 different drugs. And at least in a collaboration we have that's now extended to 100,000 drugs, but the stuff I'm showing you is just from 1,500 drugs, still quite a lot. And for each one of these drugs, they identified a gene profile. So just to be clear, you then have a drug and you know every gene that that drug affects, and you know whether that gene goes up in its expression or goes down in expression. That's on the left-hand side of the diagram. If you have a gene signature of a disease, then it's really simple. You can go online and you can find the opposite gene signature in the connectivity map, and that should give you a drug. So of course we think we know what the gene signature of Alzheimer's disease is. Maybe this is something we could enhance. I've got some ideas how we might enhance it in EPAD, and hopefully you might have some ideas of how we could enhance this gene signature and find a new drug from EPAD data. Anyway, we've took the gene signature that we thought we'd found, and we looked for drugs that are anti-correlated. That is, they cause the opposite gene expression profile, because they then should be drugs for Alzheimer's disease. Next slide. So we did that study. We screened, we screened 100,000 compounds in a collaboration, found 30 compounds that we took through secondary screen, and we now have some compounds that we hope to take through animal studies. Next slide. And instead of this costing £500,000, it cost about £50,000, and instead of taking 4 years, it took about a month. So big data analytics speeds the process. But is it any good? Next slide. Well, remarkably, maybe next slide. Remarkably, when we looked at the drugs from the connectivity map experiment, some of the results were the same as the experiment that we did laboriously and cost us £500,000 and 4 years. Some of those drugs that were the same in both studies were drugs that are used in people to combat the inflammation of rheumatoid arthritis. So then we went to primary care records in the UK, and we found, we looked at some 10 million individuals, and we looked for incident new cases of rheumatoid arthritis. And then we looked for new cases of rheumatoid arthritis where people had been given the newer drugs that tackle that inflammatory disorder. We found nearly 4,000 users, and we paired them to non-users, and we found a substantially reduced risk of dementia with a drug called methotrexate. That's the red line in the Kaplan-Meier plot that I'm showing you now. The blue line is the control group that were not treated with methotrexate. So this was a great result, and I can tell you in unpublished data, we've now replicated that exact result across EMIF, across Europe, using EMIF in 15 million records. So that was quite a lot of information, but to recap, we first used informatics to find a gene signature of Alzheimer's disease, and then we used big data analytics to establish a high throughput screen. We ran that screen against some drugs, and we found some potential drugs. We then did an online in silico screen, and some of the drugs we found were the same as from our high throughput wet lab screen, and some of those drugs are anti-rheumatoid arthritis drugs. And we did a proof of concept study entirely in silico from primary care data in the UK, and we replicated the result in silico, and then we've gone across Europe in more than 15 million people and again replicated that result. So we think we've made a substantial advance towards finding a drug for Alzheimer's disease. Next slide. However, that study, you could say, well, you found an anti-inflammatory drug, and we already knew that anti-inflammatory drugs should be useful in Alzheimer's disease, and those particular drugs had already been suggested by others anyway. What about something entirely novel? I'm going to give you an illustration now of an entirely novel a study using big data analytics. Next slide. So in this study, we first again approached genetics data, but instead of just looking at genetic data developed for Alzheimer's disease, my colleague Alejo Novardo downloaded every genetic study that has ever been done of a suitable size for any disease from the GWAS catalogue. So it's huge volumes of data. We can't analyze all of those SNPs, and anyway, why would you want to? Because a SNP doesn't cause a disease. A gene causes a disease, and maybe actually it's not a gene, but a pathway. So what Alejo did was to turn all that SNP data into gene data, and then turn that gene data into pathway data. Next slide. He then did a clustering analysis where he said which of these pathways cluster together. And you can see this is just one very small portion of that clustering analysis. He saw a number of different clusters of diseases that genetically at least seem to be similar. And this was the biggest cluster, and it contains some disorders that we don't think of as going together, like seasonal allergic rhinitis, which is a worry for me because I get that. Rheumatoid arthritis is there again. Systemic lupus erythematosus. So studies, these are inflammatory disorders, maybe not that surprising that they go together, but in the cluster or associated with the cluster, we also see Alzheimer's disease, age-related macular degeneration, and type 1 diabetes. That's really interesting because diabetes is a risk factor for Alzheimer's disease, and age-related macular degeneration is the only other beta amyloid disorder. So it has in the retina of the eye the same protein or peptide in pathological deposits as we see in plaques of Alzheimer's disease. So we seem to be finding a cluster of disorders where we have good reason to think they should be clustered, but this is just driven by the data. Next slide. So if these disorders have a shared pathways, then those pathways should be important in disease. But the problem is they have lots of pathways that are shared. So what Alejo then did was to say, well, the pathways they share, some of them will be important in relation to Alzheimer's disease and some won't. So he then said, well, the pathways that are important to Alzheimer's disease, they give different disorders, and those disorders should be associated then with Alzheimer's disease clinically. So he went to a electronic health record. Actually, it's more of a self-report claims-like data set from the US of over 6 million individuals, and he looked for disorders that are comorbid with Alzheimer's disease. And on the vertical axis here, you can see how comorbid a disorder is with Alzheimer's disease. So you can see in the graph on the left, some disorders, in this case psoriasis, turns out to be quite comorbid with Alzheimer's disease, whereas other disorders such as lupus are not so comorbid with Alzheimer's disease. And on the horizontal axis, you can see how associated these different disorders are with different pathways that are driving that clustering I've just showed you. And to cut to the chase, JAK-STAT seems to be most important. So the more comorbid a disease is with Alzheimer's disease, the more often it occurs with Alzheimer's disease, the stronger association that particular disorder has with JAK-STAT signaling. Next slide. Not content with that, we then went to a whole variety of different data sets. Most of these are open source data sets, and we said, well, if we look at the genes on the JAK-STAT pathway, are they actually altered in Alzheimer's disease? So I'm showing you here a table on the left-hand side, looking at gene transcription in blood in the AD-NeuroMed study, and then you're looking at gene transcription in blood in another independent study. Then you're looking for post-mortem brain expression study, and then you're looking at an in vitro model of Alzheimer's disease. The results are not all exactly the same, but you can see a pretty strong association with these genes. These genes are genes that are associated with JAK-STAT. These are part of the genes on the JAK-STAT signaling pathway in Alzheimer's disease. So I've gone into power saving mode, so bear with me just a second. So we look as though we are replicating this result. Next slide. And we then, you can't avoid doing some actual experimentation, so that we then went to in vitro studies. We did some in vivo studies in mice. To cut to the chase, we replicated those results and we showed JAK-STAT signaling to be altered as a consequence of amyloid. And in particular, we're seeing some what look like strong targets for drug development here, especially JAK1, JAK3, and TYK2, all part of JAK-STAT signaling. And that wasn't known before we did these experiments. We did these experiments super quickly. It took maybe 6 months to 1 year, including the experimentation or the empirical studies, and we now have an entirely novel drug discovery program based on this big data analysis. Next slide. So I've just given you, that's just a summary and a nice picture of Aleko, who did almost all of this work single-handedly. Next slide. So I've just given you 2 examples of how we're using big data analytics in phase 0 or discovery. What about phase 1 or safety? I'd like to quite strongly say here, this is not the place for big data analytics. When you're looking at how safe a drug is the first time you give it to humans, you really don't want to guess at that. Actually, there is quite a lot of big data analysis of the compounds that goes ahead even before we've given the drug to a human, and you can get some way with that, but ultimately you really do want to do those studies in humans. Next slide, I mean. So I'm now going to give you some quick examples of other phases. And for phase 2, next slide, I'm going to choose EPAD, because EPAD is a great phase 2 study. Let's go through the build on this slide, if you wouldn't mind. So next build. This is just to explain about an adaptive trial process next. I'm not going to go over this for you because as EPADistas, you understand very well what an adaptive trial is. But just to point out that an EPAD, we are in effect doing a big data analysis study next, because an adaptive trial is a complex process. So going from readiness cohort to the trial or the longitudinal cohort study, right the way through to the appendices that we'll have in our clinical trial and through to the iteration thereafter, that is a good example of a big data study. It uses Bayesian statistics, and we wouldn't be able to do EPAD without the modelers, and especially without Scott Berry and his team, Brian Tom and his team. These are big data analyses in and of themselves. Next slide. But the EPAD was set up to partner with EMIF and use the cohorts, the integration, the registry, and you may well have heard, you may be involved with, you may have heard Craig and others talking about the process that is used in the balancing committee to select people to enter the readiness cohort. Great examples of big data studies. But here I want to point out that if EPAD is to work, we're going to need to do something more than what we're capable of doing at the moment, and that is we need biomarkers. Next slide. We need biomarkers in particular to select people for our from the longitudinal cohort study, but also select people for the longitudinal cohort study more effectively than we're currently doing now. Next slide. So from symptomatic therapy, next slide, to secondary prevention, next slide, we give ourselves a problem because it's hard to find the right people. The only way we can do that is with selection and stratification markers and then progression markers. And I'm going to talk about selection and stratification markers now. Now you could argue, next slide, that we have all of the selection markers we need in CSF or in PET imaging. And we are, of course, using these in EPAD, but let me show you a problem. Next slide. The problem is one of cost. So here are two examples, one from a study I'll tell you about in a moment that has just 250 people. The other is for the proof of concept trial. Let's say we want to do a proof of concept trial in EPAD with 1,500 people. I've estimated the cost if we select by PET imaging. The screen failure cost alone for a study of 1,500 people is 2.7 million. If we were to include tau PET as well, it goes up to an alarming 7.2 million. That's the screen failure cost, not the cost of doing the trial. That's the money you have to count on before you even start the trial. I think we need a better marker to select people that will reduce the screen failure cost at PET imaging. I think we may have one actually, but if we look at the potential, I think that even if we had a bad blood-based biomarker, we could reduce the costs by 1.9 million to a little under 1 million screen failure cost. That would be a huge saving for EPAD and studies that follow it. Next slide. So how best to do that? Well, to do that, we used EMIF. I wanted blood samples from people that have CSF, A-beta and tau measures. Conventionally, I'd go out and collect a new cohort. That takes typically 5 to 8 years from the time you have the grant application to the time you have the samples, and cost, and I know this because I've done it, a little over 10 million euros. We did it in EMIF. I was going to say for nothing. Actually, it wasn't nothing. It did cost us something, but about 100,000 euros as opposed to 10 million euros. And this is the true time it took from the time we had the first idea. It wasn't zero time. We started in June 2014. We finished the study in June 2018, including all the wet lab analysis. So it took 4 years as opposed to 10 years. But most of that time was taken up with contract negotiations. So some great work from Synapse and our other colleagues, but there's a great potential to speed up this process even further. Next slide. So what we did, we used the cohort. We went to the cohort. We found out from the catalogue whether the cohorts had the data, including the samples that we wanted. We found out from the catalogue how to approach people. We approached them. We got the samples. We did the analysis, and I'm showing you for the first time here the results of that analysis. So when we look in the whole new cohort that we established from EMIF, this is from 1,000 or so people, we found 7 proteins that can predict whether these individuals have high amyloid load or not, with an area under the curve of about 0.75 that compares to age only of 0.63. Importantly, this wasn't driven by ApoE genotype. We can look in the ApoE negative people and get more or less the same results. So this is gold dust for the pharmaceutical industry. It's exactly the kind of blood-based biomarker that we need, and we're now moving towards trying to put this onto a assay type that would be viable for very, very large studies. So we were able to do this study at low cost in a relatively short period of time using the EMIF catalogue, and I hope that before EPAD ends, we'll be seeing this blood-based biomarker used to enhance the screening process. Next slide. Very briefly then, I'm coming up to a close in phase 4. Next slide. After a drug is released, we can use the electronic health records to see whether a drug does any good in the real world. What you're looking at here is data from a single hospital in the UK. You're looking on the vertical axis at MMSE score from people that are declining. The first inflection point, time 0, is when they're given donepezil, and you can see people get a little bit better for about 6 months, then they get worse. That is exactly what is found from the pivotal trials. It's what we expect. The difference is this came from large numbers of people. Actually came from what you're looking at here is 8 times more data than has been produced in every single research study that has ever been done on donepezil, and it came from analysis of electronic medical records from a single hospital. Next slide. So the drug does, next slide, the drug does what we think it should do, next slide, once it's released in these 3,075 individuals. We can also look at some other things. And the important other thing that we looked at in another study, next slide, was what happens to these people. So we took 6 months windows from people randomly selected, and we extracted using natural language processing what happened to these people, what kind of resources they were using in the period after their index date. Next slide. And we were able then to work out the costs of their care provision and showed that these costs were associated with severity, living alone as you'd expect and so on, but not with physical illness, which was unexpected, or depression or gender. So in this study, that 2 studies in fact that I've spun over very quickly, we're able to use big data analytics to show that the drug works in the real world and also provide the data that the people who have to pay for this drug need in order to give authorization for payment, whether that is the NHS and National Institute of Clinical Excellence, NICE in the UK, or the payers in your own country. Next slide. I've missed something out. I've missed out phase 3. Next slide. Phase 3 is interesting, and I'm going to show you a quick example of how we might in the future use data that comes with real velocity. So phase 3 is an obstacle to progress. I don't know whether you all know this, but the cost of doing a phase 3 trial in Alzheimer's disease is approximately €1 billion. Honestly, there aren't many companies in the world that can afford to lose $1 billion on a clinical trial, and of course every clinical trial has failed up to now. So we need more effective, very large scale clinical trials. We need to make recruitment quicker. We need to have outcome measures that are better. Next slide. So could we recruit people for clinical trials more effectively? I think we could from electronic medical records. So this is what we're now just beginning to do in the UK. Next slide. Using the CRIS system, the electronic health records system I told you about, first of all, everybody that comes through a trust that is using this system, everybody gets consented for us to go back and it's called consent for recontact, to go back and contact them again. Next slide, for research. So what happens, a researcher in silico using the record identifies somebody that might be suitable using pseudonymized data for their clinical trial. Next slide. There's then a process of matching IDs in a trusted third-party system. Next slide. If the patient has given their consent, then they are re-identified in the electronic medical record. Next slide. And the doctor or the researcher then goes to the patient that you want directly. This speeds the process. Not only that, but the process is accompanied by a system so that it's automated, and the researcher gets contacted by email every time a suitable patient enters the system. Next slide. Patients agree to this. So when we measured whether patients agreed, 74% of them said yes, of course. Next slide. We also need to collect data on patients better, more effectively than getting them to come back to the clinic for a complicated one-day assessment as we do in EPAD right now. So with my colleagues, we're working on using connected devices, next slide, basically a phone, for measuring cognition, movement, and all sorts of other data that might be surrogates for behaviors and cognition relevant to dementia. If this works, then it might be we could measure people not just once every few months as we do in EPAD, not just on a day assessment as we do in EPAD and every other study, by the way, but we could do it more or less continuously, and we could do it at scale for nearly no additional cost, identifying when individuals change or become appropriate to call back to the clinic for reassessment. This would allow us to scale phase 3 trials very considerably at very low cost. Next slide. So I think that trials in the future are going to look very different, and we're going to be using drugs that we've developed in silico. We're going to be doing proof of concept in real world data using biomarkers that have been established using big data analytics. We're going to do phase 3 trials where we recruit people in silico and we do some assessments in silico continuously and at huge scale. It doesn't mean we obviate the need for a human intervention and assessment, but it does mean those humans can be used for things that humans are really, really good at and leave the machines to do the rest. Next slide. Next slide. So I'm not going to dwell on this. I think that we're in an interesting time of big data analytics. I hope that EPADistas and the Academy in particular are interested in getting stuck into this and learning some of the techniques of machine learning, artificial intelligence, and multivariate statistics on very, very large data sets that I've just been talking about. I'd be delighted if any of you wanted to come to Oxford or to any of our other sites that use these techniques, and we'd be delighted to help you progress this if it's something you're interested in. Next slide. We've got some, next slide. I should remember to say there are many other platforms, including Global Alzheimer's Platform and GAIN in the US. Next slide. I want to thank some of my colleagues, especially in the EMIF teams, the CRIS teams, the EPAD teams, all working seamlessly together, and my informatics team at Oxford that you can see there. Next slide. I just want to, you may not have seen this person. This was a British parliamentarian, an MP. Her name was Tessa Jow. She was a very good MP. She died about 8 weeks ago of a brain tumour. And in the last 6 months to 1 year of her life, she became a massive advocate for two things that are very relevant to EPAD. First, she became an advocate for sharing of electronic medical records, and she made all of her own personal data available to researchers. And she also became a powerful advocate for adaptive trials. And she was on the British television, radio, and here she is speaking in the House of Commons about the importance of data in medical research, and specifically about the importance of adaptive trials. That led to an injection of money, £50 million, to adaptive trials and big data research in brain tumour in the UK. But I think what she did to socialise the concept of big data analytics, sharing of electronic medical records, and specifically adaptive trials was very important. And although she was doing this for brain tumour, brain cancer research, I think those of us that work in this area, in EPAD in particular, also have a lot to thank her for. I've gone over time, I'm sorry, but there is, I hope, a little bit of time to talk about big data analytics and data saves lives. Thanks. It's been a pleasure. Thanks, everybody. Bye-bye.